Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
Go above and beyond.
they become very good at what they do, but only
0.1%.
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
With Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Irvin C. Gerling. He's a professor of medicine and endocrinology at University of Tennessee Health Science Center, and he's doing some type 1 diabetes research. So we're going to talk about that. Ivan, thanks for coming.
Well, thank you. I'm glad to be here.
And I see in your bio that you received a large grant recently from National Institute of Diabetes and Digestive and Kidney Diseases. So that's great. I hope that that funds your research and it goes well.
Yes, we are actually quite a bit into that project already.
Okay.
And essential for any research is that you have to find funding for it. So it's always a relief every time we get another grant.
Excellent. Well, tell me about your research, and then maybe tell me about, if you can, the specific grant focus of the research.
Sure. So I'm working and doing research in type 1 diabetes, or also called insulin-dependent diabetes. Others have called it juvenile onset diabetes. It tends to be occurring in young people, and it is usually not associated with overweight, but rather it's associated with what we call autoimmune disease, which is when the immune system, which usually helps protect us against bacteria and viruses, fights off infections for us, when it suddenly makes a mistake and begins looking at the insulin-producing cells in our bodies, which are called the betacells, looking at those as foreign and starts destroying those cells, even though they are not foreign.
So what's your particular angle of attack here in the research?
So we have for a long time been well aware of that the immune system is doing something here that it shouldn't be. What we not have had less understanding of is that why exactly does the immune system make this mistake? The immune system is usually very good at deciding what is foreign and what is not foreign, and it attacks those things that are foreign, and it leaves our own tissues alone. So why is it that in some children and young people, the immune system makes this mistake and says, okay, these insulin-producing beta cells, I'm going to destroy them. And there's sort of two possibilities. One is there's something wrong with the immune system, but there's also the possibility there's something wrong with those cells. So a lot of work has been done studying the immune system and what is it that goes wrong and how can we try to block those things. But perhaps less has been done about looking into, is there something wrong with these beta cells in these individuals, and is that why the immune system makes this mistake?
It seems like there's a latency period between birth, if people are born with this. Do you think they're born with it, and there's just a latency until the immune system turns on the beta cells, or does it develop for some other reason and it's not necessarily innate? Well,
there is. We know from studies in monozygotic twins that even there, when you have a complete identity genetically, there's actually just slightly less than a 50% concordance rate. So it's not always that two completely identical, genetically identical twins get the same disease. So there are certainly some environmental factors that seems to be playing on a genetic susceptibility. And if you are unlucky enough to have a sufficient number of predisposing genes and you encounter an environmental trigger, then we get this autoimmunity going.
So any idea on what the very beginning of the condition looks like? Later on, I'm sure we know more, but has anyone looked longitudinally at what happens in the pancreas and what other
Well,
downstream effects or comorbidities occur?
yes, so it's very difficult, of course, to study this in humans. We have some decent animal models, but there are also a lot of differences between animal models and humans. So one thing that really has helped has been Juvenile Diabetes Research Foundation set up this organization called NPOD, and they are collecting organ donations from humans who either have diabetes or are at high risk of getting diabetes, and then, of course, also control issues. So actually studying what happens in the pancreas of a human being has not really been possible until recently. And it has been with this resource that it's been possible to really make big studies, and a lot of different investigators are looking at those same organ donor tissues, studying different things. It probably is something may happen early, way before you become a clinically type 1 diabetic patient. There is autoimmunity going on. And we have been able to study people looking at their bloods and looking for signs of autoimmunity, and we have found that those things can occur easily 5-10 years before you become diabetic. So there's a long drawn process of disease. But what I've been interested in is what is it that provokes the immune system? The very first provocation before you have any autoimmunity going on, what's going on at that time? And we have been able to use these organ donor tissues to study and see what is going on there. And we have seen indications that there is stress in this tissue, even way before anyone would have become diabetic, and just at the time when they have maybe one autoantibody, so one little indication of autoimmunity, there's stress in their tissue.
What kind of antibodies are produced, and do they change over time? Like, what's been observed?
So the antibodies we are using to diagnose autoimmunity to beta cells are antibodies that basically are specific for beta cells or only occur in a few tissues, including beta cells. So that's what we are seeing in the serum of individuals. And we've done family studies and looked at someone who's at risk because their family have type 1 diabetes and they have these high-risk genes. They've been followed year after year after year, and we're seeing that they will develop these autoantibodies, indicating that there's autoimmunity to their beta cells. Of course, insulin is the major hormone produced by the beta cells, and it's the lack of insulin that gives you diabetes. And we can see insulin autoantibodies develop way before someone become diabetic. There's a protein called glutamic acid decarboxylase. So there are several different types of antibodies, and the more you get of them, the more likely you are that you will become diabetic, and the sooner it will happen, the more antibodies build up.
So over time, you see an increase in antibodies. Do you see the appearance of new types of antibodies, or they stay the same throughout the whole course of disease?
It tends to be that if you develop one specific antibody, that will stay there until the time of diagnosis. You may then pick up another antibody, and you may pick up a third antibody. So you get one and it sort of stays there. It might go a little bit in and out, but it tends to be there throughout the pre-diabetes stage. And then eventually when you become diabetic, it's because most of your antibodies or most of your beta cells have been destroyed. And after diagnosis, a lot of individuals then lose their antibodies. And it probably is because there simply isn't much of anything to stimulate the production of these antibodies after most of your beta cells have been destroyed.
Have people been able to look at the structure of the antibodies and then back calculate what caused, what would bind to the antibodies? like the antigens themselves, do we know what they are that are either presented on the cell membrane or given off by the beta cells?
Yes, but interestingly enough, we actually don't think the antibodies are as important for the pathology, but they are very good markers. Probably most of the destruction is coming from another part of the immune system, the T lymphocytes. But they have been really good for epidemiological studies because they do very well seem to indicate something is going wrong inside this person's pancreas. And it's not possible to take biopsies from a pancreas. It's just too dangerous. So the only way we can really follow what happens in the pancreas is by these indirect biomarkers in the form of autoantibodies. But we kind of then in my work, where I'm more interested in what exactly is going on in the beta cells, it's been really good to have these organ donor banks with large numbers of pancreas available for our studies.
So, okay, so you can't do biopsies, so I guess you'd have to get cadaver beta cells. Yeah.
That's the only way we can do it, yes. It is, unfortunately, taking a biopsy from a pancreas, it carries a number of risks to the individual, so it's not something we can do.
I thought you could, some people have pluripotent stem cells. They can differentiate into beta cells to study them.
Yes, and there are also beta cell lines, but it's very difficult to get those to really mimic what's going on in a whole human being. So we can get some information about beta cells and how they respond to different things using cell lines, some derived from stem cells and others from tumors, etc. But we really kind of like to study these cells in their natural environment, and that means either going to animal models or try to get organ donor material.
Okay, so I have a little bit better picture now. So what specifically are you trying to pick apart and figure out, if you could restate it?
So we have had for many years the idea that maybe certain viruses would be involved in creating this stress in the beta cells, and that's what initially get the immune system to be interested in the beta cells and then start producing this unfortunate autoimmune destruction of the beta cells. So there's been some epidemiological studies indicating that maybe there's a connection between viruses and initiation of autoimmunity, and there's even been animal models used. But we have kind of said, well, we need to look at stress in islets, in the beta cells in the islets of Langerhans. So we want to collect these islets from people who are at high risk of becoming diabetic, seem to be in the early process or in the late process. So what are the overall gene expression signatures? And the novel technologies allows us to basically take out a piece of tissue and get an answer on how many genes and at how high a level are these genes expressed. So then we can do comparative studies, and we can say, well, what's the difference between an islet that seems to be under immune attack and one that is not? And are there in individuals where we clearly have initiated the process, does a normal-looking islet look different from a normal-looking islet in an individual where we have no indication at all that there's a process going on? So that's what we've been doing. We have been taking this tissue, then we have cut out individual islets, and then we have looked at their global gene expression patterns and then tried to connect differences in global gene expression patterns with indicators either that of disease, quote unquote, in the islet itself, or with no knowledge about the organ donor that there might have been initiation and early stages of a disease process.
So, I mean, what have you observed so far? Have you had access to beta cell lines and, healthy ones, and have you been able to look at the differences?
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
Yes, and we have actually found, interestingly enough, that does seem to be some indications in the individuals that have early markers of a disease process, that they're indications of viral infections. So tissue will normally respond to a viral infection by turning on a number of genes that are effective at trying to fight off a virus. So we have sort of that footprint of a virus by looking at the tissue's expression of certain stress genes. And some of them are very specific to specific types of infection, and some of them are sort of generalized infection markers. So we have found both generalized infection markers as well as some that are very specific to double-stranded RNA viruses. We found these indicators both at the very early stages of autoimmunity and also later stages of autoimmunity. And when we look at the islets that look completely normal in a person that had no indication at all of autoimmunity versus those in individuals that have very early indicators of autoimmunity, we are seeing a difference in these genes that would suggest that there might be a close correlation between the disease process and responses that are associated with viral infections.
Okay, is it so you're seeing differences on a genetic level, which would seem to say that a person is born with a propensity towards this condition, or are you seeing also epigenetic changes? So you're looking at methylation and other marks?
So we've not looked at the methylation, but we do know that there are genetic susceptibilities to this disease. So individuals with certain genes are much more likely to develop this disease than individuals with other genes. And there's been family tracing studies to identify with what genes, when they are transferred to the next generation, which one of these genes seem to transfer the susceptibility to the type 1 autoimmune diabetes. So we know a fair amount of that, and we know that there are amongst these that the very strongest genetic association is with immune response genes, the HLA complex in the human genome. If you have certain specific variations, genetic variations in that in those genes, you are much more likely to develop autoimmune type 1 diabetes. And there's also actually a gene that is associated with type 1 diabetes genetically with susceptibility. That specific gene we have found to also be overexpressed in the islets of Langerhans and in the beta cells there in people who are in the early stages of these diseases.
So when you say you see genetic differences, does that mean that the genes are upregulated to start? Or
no,
maybe they're more sensitive to upregulation or overactivity?
We don't know for sure, but the general genetic susceptibility is basically the base pairs, the sequence of that DNA that all of your cells have. So there's variations in that, and we can sort of follow specific variations where the code has been changed one or two places, and we can say, well, is that change associated with being more likely to become a type 1 diabetic? Then we can also, of course, look at gene expression levels, and that's what my study has done, is to say, okay, when we look in the islets of Langerhans and in the beta cells in the islets of Langerhans, are there certain genes that are higher expressed? And that's what we are looking for in my study. And we have looked and found that a number of genes that are associated with responses to viruses, that they are upregulated. So they are expressed at a higher level in people that are in the early stages of developing type 1 diabetes.
Has anyone looked at people with type 2 diabetes and sequenced the islets to see if there's similar genetic changes, or again, even epigenetic changes at that time, maybe pancreatic cancer?
We have actually also, as a control for comparison, we have also looked at islets from individuals with type 2 diabetes. we have to do that because some of the gene expression changes may simply be because of blood sugar fluctuations. So there, the type 2 diabetics would sort of help us define what are the genes that are changing because of blood sugars and what are the ones changing because of autoimmunity. So yeah, we have looked at that and we have found that there are some other types of changes in type 2 diabetes than in type 1 diabetes.
Oh, and can you correlate whether there's changes in, again, related to relationship to the immune system or just because of the sugar load or the insulin load, the demand for production, that it changes the cells in a different way?
Yes, so that's what we use those. So we have islets of Langerhans and beta cells from normal people with no diabetes, no indication whatsoever of autoimmunity. Then we have from individuals that have indication of autoimmunity but no diabetes. Then we have from individuals that have autoimmune type 1 diabetes. And then we have from individuals that have non-autoimmune type 2 diabetes. So with those groups, we can start sort of sorting out what is because of diabetes or blood sugar fluctuation and what is because of the autoimmune.
Does there appear to be any immune response in type 2? I know that's not your field, but is there any correlation or?
We are not seeing much at the level of the islets. We do know from other people's studies that there is probably inflammation is associated with type 2 diabetes, but it's by another mechanism. It's not affecting the islets directly. It's basically causing what's called insulin resistance in tissue like muscles and fat and liver. And that's part of what is causing type 2 diabetes, where you still have plenty of beta cells, and in some patients, even more than normal numbers of beta cells. It's just that they produce insulin that just doesn't work because of what we call insulin resistance in these type 2 diabetics. And it looks like inflammation is causing insulin resistance.
Well, I've heard insulin actually as I guess I don't really know if it's a protein, but it appears to have a folding component in order to make it functionally effective. Is there any misfolding of insulin when beta cells are under high demand for production or in type 1? And could that be identified as something attackable by the immune system?
Yes, absolutely. There's definitely, when a cell comes under stress, doesn't do as well producing good functional proteins. And insulin is a protein. It's a peptide protein that's secreted into the circulation, and then it goes out in the tissues via the blood and does its thing. And yes, there is certainly, when you put more and more stress on a beta cell, it produces more defective, deformed, inefficient insulin. And there's certainly reasons to believe that those proteins, those defective insulins can become targets for immune responses. They might be oxidized or other things by the inflammatory processes in a stressed beta cell.
Yeah, what happens when a beta cell is stressed? You said it doesn't do well, but what does that mean? Does it create defective insulin? Does it not convert proinsulin properly? What happens? It
probably doesn't process as well. So there has been studies suggesting that we get more proinsulin released. And proinsulin is sort of a unprocessed product of the insulin gene. So stressed beta cells seem to be releasing a higher proportion of proinsulin, which suggests that they are not quite capable of doing the job. There is probably some of the problems with when we get autoantibodies to insulin. They might have initially originated from proinsulin autoantibodies.
So if you go further in the gene discovery, what is this going to tell you to do? What kind of therapies does it suggest?
We're looking into what are the stress pathways that seem to be activated when these beta cells start going downhill. And if we can identify that, we would have pathways that could be targeted with specific drugs. So if we can fully understand some of these stresses, maybe we can design drugs that can help the stressed cells by biochemical means. So that's certainly is one pathway whereby this could end up in treatments helpful for patients. The other thing is if it turns out that a large number of individuals that the first steps are viral infections, maybe if we can identify some of these viruses, we can end up with vaccines for them. And there is already a study in Europe that is trying to come up with a vaccine for a specific Coxsackie virus that is believed to be a possible precipitant of type 1 diabetes. So those kind of things, if we can show that this is really where the whole thing starts, then we can make the argument that maybe we should develop vaccines to try and help protect individuals so that they never get this autoimmune process going.
Well, if you look at the stress hypothesis, maybe that's why there's a latency period that's variable for different people. People that, starting with what you found, their genes are different. They're predisposed to type 1. Maybe they're, the beta cells are more subject to stress, variance in glucose levels and other factors. Therefore, they are stressed to a larger degree and earlier on, and they collapse. So they revert to this state that triggers autoimmunity. And maybe that's maybe that's how the cascade happens.
Yes, yeah. I mean, there are a number of hypotheses, and we are trying to sort of look and create data, either supporting or rejecting these many different possibilities. But we probably are also looking at a heterogeneous disease pattern. It's possible that a number of different types of stress all could lead to precipitating the immune system attack on the beta cells. And there are other groups that are trying to find out, if we can't stop it from being initiated, can we at least find some way of blocking it from progressing? Because you don't get type 1 diabetes until about 90% of your beta cells have been destroyed. So there is some room there for coming up with interventions so that an individual will never actually get the disease.
Is there beta cell destruction in type 2?
Yes, but it's different. In type 2, initially, your insulin doesn't work so well, and your body is responding to that by expanding your beta cell mass and producing more insulin. So your insulin levels get to be abnormally high, but that's your way of compensating for your insulin resistance. But down the road, and some believe maybe in some people at a very early stage, you also have deficiency in the beta cells, and whether it's individual beta cells that don't work so well, or maybe it's the beta cell mass that if you need, let's say, 10 million beta cells and you only have 6 or 7, that would make it harder for your beta cells to handle the top load right after you ate something very sugary. And that could then create some stress in those cells. They might die simply from the stress, not from the stress-inducing autoimmunity.
So in looking at, cadavers or, and the beta cells, you don't see oversized beta cells. You just see they're just not there. Or do you catch any that have just recently died, and can you tell what happened? So
yeah, when we look in these type 1 and high risk of type 1 donor, we are seeing that the insulin staining cells have disappeared from this islet-Langerhans structure. So there are a number of different cells in the islet-Langerhans. There's a lot of insulin-producing beta cells, but there are also glucagon-producing cells, alpha cells, and some other types of cells. And in type 1 diabetes, it's very specifically the beta cells that produce insulin, they are gone. Depending on the stage, you may find that they are gone from only a very small number of islets, or they are gone from a very large number of islets.
That's interesting. Do they leave behind any scarring, or are they like ghosts and they just were never there? It looks like
it basically is just that they are not there. So you can go in, you can look at the tissue, you can stain it for presence of different hormones, and you are basically seeing that this is an islet, and it doesn't really stain for insulin at all. It does stain normally for other hormones like glucagon, but it's just somehow the beta cells either have been diverted, turned into other types of cells, or they have just been apoptosed and disappeared without leaving a scar.
Yeah, that's kind of odd. It's like a mystery, they're gone.
They're gone, yes, that's for sure. And we are very sure that it's because of lymphocytes that have gone in there. We don't know for sure to what extent. It also may be because there are processes that stops the production of new beta cells. So there might be several processes going there, both the fact that beta cells are being destroyed by apoptosis due to an immune attack, but it could also be that eventually the ability to keep up with that destruction and replicate and make new cells, that is somehow being hampered.
Last question, is there any way to non-permanently stain or attach fluorescent tags to beta cells inside of someone? Can they drink some kind of dye that preferentially goes to the beta cells in the pancreas so you can see them?
Yes, there are actually some interesting work being done by other groups, including one at Vanderbilt, trying to put in dyes that would stain the beta cells, and then using scans to try and measure how much beta cell volume is in the pancreas of a living human being. And they have some preliminary data, and that actually produced some interesting new insights. It turns out that there's probably a lot of difference in how much beta cell, how many beta cells individuals have. And some may have 10, 50 times as many beta cells in their pancreas as others. And then we're talking about individuals who have not become diabetic.
Right.
Big natural variation seems to be present.
Well, very good, Ivan. This is really fascinating. What's the best way for people to find out more about your research?
Well, I would say just look me up. You can always find, I think nowadays, even Google will help you find scientific articles, and they can learn more about my work that way.
Well, very good. Ivan, thank you for coming on the podcast. I appreciate it.
Well, thank you for inviting me.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.